Next up, we have Luuk Harbers, who's a PhD student in the COVseq and focuses on method development in cancer genomic. And Luuk will talk to us about genomic surveillance of SARS-CoV-2 using COVseq and making it fair. So please go ahead, Luuk.
Yes, thank you. Yeah, so as been mentioned, I will be talking about genomic surveillance of SARS-CoV-2 using a method we call COVseq and how we try to make it fair. So yeah, normally we don't do any research on viruses or anything. We mostly do cancer genomics. So briefly, one slide of background. So for the purpose of cancer genomics, we developed a method called COVseq, which is a method for high throughput sequencing of multiple, for instance, cancer samples in parallel. And how this method looks like briefly is you have extracted DNA. This can be tissue, this can be FFPE tissue sections from cancer samples, basically anything. You have one sample in each well of either a 96 well plate or 384 well plates. You have a step called restriction digestion, where you digest the DNA with a restriction enzyme. Following this, you ligate adapters to this. So in each well, you have a specific sample, specific barcode. Then, because every sample has its own barcode, you pull everything together. You have an in vitro transcription step, reverse transcription, PCR, and then go to sequencing. So that was a method we developed in the end of 2019. And then, of course, the pandemic came. And so we decided to try to adapt this method to also be able to sequence many SARS-CoV-2 genomes in parallel. So that's what we did. And we called this method COVseq, which was published in Nature Communications, which I worked on together with Michele Simonetti and Ning Zhang. So this COVseq workflow, I will just show the workflow, a few slides of brief results, and then go to the FAIR principles. So because we don't work with extracted DNA, but we work with extracted RNA from COVID patients, we first started with a step, Multiplex Pcr Step. So initially we used the CDC protocol for this multiplex PCR. So again, we have 96 multi-well plates, or we have multi-well plates of 96 wells or 384 wells. Each well has a sample. And then we have 6 plates for 1 plate per pool, basically. So we do a multiplex PCR, and basically that's all we do. And then we go into the COVseq workflow that I described previously. Another key difference is with COVseq, for the restriction and digestion step, we use 1 restriction enzyme. Here we actually decide to use 2 restriction enzymes at the same time. We included MSC1, for instance, because here you can see the locations of the restriction sites, and MSC1 has a lot of restriction sites inside the spike region of the SARS-CoV-2 genome. So this increases the coverage in this region. So briefly, a few results. So initially, we obtained 29 SARS-CoV-2 samples. We decided to sequence these 29 samples, both with a commercial available library preparation protocol called NebNext, and then also with COVseq. And as you can see here is that there's a high correlation in the number of reads we obtain both with NebNext or with COVseq. And the number of reads you obtain is highly also correlated with the CT value of the sample. So the CT value basically gives you information about how much SARS-CoV-2 RNA is in your sample. So a high CT value means that there was very little RNA in your sample. You can see here that these high CT value samples get very few reads, both in NebNext and in COVseq. Next, it's a similar plot, but instead of the number of reads, we show the 10x breadth of coverage. So basically the percentage of the genome that is covered at least 10 times, so at least by 10 reads. And here again, you see that there's a high correlation, and the low CT value samples here in the top right, they basically get near 100% breadth of coverage at 10x. But there's then the higher CT value samples here that obtained way fewer reads, so also have a lower breadth of coverage. But here we see that actually COVseq is more sensitive here and actually has a higher breadth of coverage compared to NetNext. Then we want to see if all these reads we obtain, if they only align to the SARS-CoV-2 genome or also, for instance, to human. And we saw that here on the right are the samples with a relatively high CT value, so very little input RNA. We see that there's actually a lot of the reads, a high percentage of the reads, not only or not at all aligning to SARS-CoV-2, but aligning to the human genome. This will be important a little bit later on when I talk about the data sharing. Following this, we obtained 95 extra samples, of which we prepared 3 replicate libraries with COVseq. And as you can see here, we did then SNV calling on these samples. And as you can see here, almost all the single nucleotide variants that were called were shared in all 3 libraries, showing a higher reproducibility of this method. And here's the heat map showing basically all these SNVs. And you can clearly see 2 kind of clusters. Here on the right is a cluster with all these mutations at these locations in the SARS-CoV-2 genome. And this cluster was basically the SARS-CoV-2 variant that was going over all of Europe, kind of the main variant, so to speak. And here on the left, there are 7 samples that actually were assigned the alpha lineage, the pangolin lineage B117, which was the alpha lineage, which was at that point when we received the samples, basically just starting to surface more and more. So we show that we can easily identify also these different lineages. Then maybe for a lot of people that want to then do genomic surveillance, maybe the most important metric is because it has to be cost effective, right? It has to be cheap. If you want to do genomic surveillance and you have to sequence a lot of samples each week, for instance, it has to be cost effective. So first of all, we further improved the protocol. Instead of using the CDC primer pools, we used Arctic, which only uses 2 primer pools and is cheaper. And then we show here basically the cost of COVseq compared to 3 different methods, CleanPlex, NetNext, and Nextera. And you can see that COVseq is just way more cost efficient. This is obviously a large number of samples, but even if you go to around from 200 samples, it's already cheaper. And then if you go all the way, it would come down to approximately $14 to $16 US per sample, compared to like over $60 US per sample. So that was the brief results that I wanted to show. So how did we try to make this as fair as possible? So first of all, of course, we uploaded all the data. So here we uploaded all the BAM files to the European Nucleotide Archive. So the reason why we uploaded the BAM files and not the FASTQ files, which is normally done a lot, is because we showed previously, or I showed previously, that in these FASTQ files there were still, especially in the high CT value samples, a decent amount of human or of reads that map to the human genome. So this then would be due to patient information, we could not upload this with open access. So we decided to align the BAM files to the SARS-CoV-2 genome and then upload these files so it could still be open access and available for everyone that was and is still doing COVID research or needs these sequences to do their research. So in total, we uploaded 691 BAM files from 274 samples that we sequenced. So there were a decent amount of replicates there as well. And here we, of course, we had sequencing related metadata, we included that, and also sample-related metadata, such as you can see here some examples: the geographic location, the host disease outcome, the host age, also how the isolation was acquired. And other than that, we also made all the code available in GitHub. And as you also heard in the previous talk, it's not just having the code available on GitHub is already really nice, but we also then, at the moment of the publication, we linked this GitHub. So we made a release and linked this release to Zenodo, so that there's always a persistent identifier linking to the GitHub release from the code that was actually used for all the analysis and data processing of this paper. And also, that you saw previously, we had in my opinion, pretty elaborate README file going through all the steps of pre-processing, running everything. We also used nfcon file recon to run all the samples. So those were the things we tried to do and try to make elaborate as possible and upload as much metadata as possible to make it as fair as possible. And with that, I want to thank everyone, of course, in the Bianco Crosetto labs and our collaborators in Italy that provided all the samples, all the COVID samples to us. And if you have any questions, I'm happy to answer.
Thank you, Luuk. Do we have questions? So, did you think about the FAIR principles ahead of time, or is it?
I mean, I don't, not necessarily with like the name FAIR principle. We did, of course, think about, okay, we need, like, how do we share all our data before? And then for me specifically, because I did most of the analysis, how do I provide and how do I like make the GitHub and all the codes available that is also useful for other people's, because I think me and a lot of other people, every time you go to a GitHub link where they share all their code, and it's still not really that useful, because there's, for instance, not a README file, and the README file just says code for this publication, and that's it. So in that sense, we did think about it, but not necessarily with all the steps of specifically FAIR in mind. I think that's fair to say.
Now, having thought more about the FAIR principles, do you think in the future you will sort of think more about these aspects?
Yeah, I think so. And also, I think every time now with new projects, you realize what you should do and what could be improved way further. So also, if I would start this project now, there's also already things regarding the FAIR principles that I would change, and I would have done differently for sure. Yes.
Well, I'm very happy that you published the data and made it available, because we reuse it in a data management course we run. So excellent, I think.